Hello, everyone. Welcome to today's Leveraging Lessons from the Past webinar, Environmental Conditions Risk Assessment of the Clinical Laboratory. This is Jenna Peterson, an Education Specialist for Mayo Clinic Laboratories. Our presenter today is Lisa Button, a Quality Management Coordinator in the Department of Laboratory Medicine and Pathology at Mayo Clinic in Rochester, Minnesota. A couple of reminders for everyone before we begin today. There will be a question and answer session at the end of the presentation. You can submit questions online via the Q&A icon here in Zoom. Please only use the chat function for any technical issues. Also, today's program is being recorded and will be posted to our website in a few weeks. And now I will turn things over to Lisa.
Great, thank you so much. I'm happy to be here today. As Jenna said, our presentation today is about environmental conditions and a risk assessment that would go into it for the way that they might impact the choices that you make within your clinical lab. I have no disclosures to announce. Our objectives for today, we're going to assess CAP and CLIA standards for environmental conditions in the clinical laboratory. We'll describe methods of determining upper and lower thresholds for temperature and humidity. You'll be able to evaluate potential risks associated with operating laboratory instruments and equipment outside of manufacturer-identified environmental conditions. And we'll review an electronic tool that was designed for laboratories to record environmental thresholds and demonstrate the use of that tool, how we're using it here. Before we get into the content of the presentation, I wanted to build a base level for all of us. So this is a terminology page, things that might sound familiar to you or maybe you've not heard before, but you will hear during the presentation so that we're all on the same page. If I say building automation system, or if you see BAS on one of the slides, you'll know that that is referring to a centralized computerized network of devices that monitor and control a building's infrastructure. Oftentimes, this is a system that's controlled by your facilities team at your hospital or laboratory. You could think of this as something that's managing or controlling the HVAC system, so heating and cooling, lighting, security, and fire systems. When I talk about environmental conditions, I'm really focused on the CAP and CLIA focus for environmental conditions. So we're going to be talking about temperature and humidity today. If I say the word excursion, I don't mean that we're going on a trip. What I mean is that it would be an instance when temperature or humidity go outside of their defined range. They've taken an excursion outside of that range, whether high or low. And then if I say threshold, I'm referring to either the upper or the lower end of the temperature or humidity range that's been defined for that instrument or equipment. All right, we're going to set the stage with what does CAP say about environmental conditions in the clinical laboratory? We have the all common checklist item 30775 specific to temperature. CAP says that acceptable ranges are defined for all temperature-dependent storage devices, equipment, and environments in accordance with the manufacturer's instructions. A new CAP checklist item that just was put into practice in their newest revision is for humidity 30580 on their all-common checklist. It states that test systems are operated within manufacturer-specified humidity ranges, and that this requirement applies only to test systems for which the manufacturer specifies a humidity range. Humidity used to be part of this next one on the slide, which was the climate control lab general checklist item, but that was retired in favor for moving it to the all-common checklist and making it specific to humidity. To refresh your memory a bit, there are CLIA regulations for laboratory environmental conditions. So the CFR, or Code of Federal Regulations, that's cited, or that's listed below, is cited in the CAP references that we just saw on the previous slide. So 42 CFR 493.1252B states that the laboratory must define criteria for those conditions that are essential for proper storage of reagents and specimens, accurate and reliable test system operation, and test result reporting. The criteria must be consistent with the manufacturer's instructions. That underline there was added by me. That's not in the CFR specifically, but I wanted to call your attention to it. So if the manufacturer provides instructions, we should be following it. These conditions must be monitored and documented, and if applicable, include water quality, temperature, humidity, and protection of equipment and instruments from fluctuations and interruptions in electrical current that adversely affect patient test results and test reports. If you're newer in your role as maybe a quality or a technical specialist, and you're wondering where might I find these temperature and humidity ranges? So commonly for instruments, equipment, reagents, and supplies that are used in test systems, you might be finding these temperature or humidity ranges in manufacturer instructions for use, product inserts or package inserts, or in the operator, owner, or service manual that comes along with that item. And then on the right side of the slide, I have a screenshot of just a common piece of lab equipment, what it looked like in that owner's manual. They had a section for environmental conditions. Oftentimes, these owner's manuals are coming in an electronic format now. So like a PDF, you can do a find function within them. I'm oftentimes searching for the keyword of humidity or temperature, and that will return what it is that you're looking for. All right, so what do these standards mean for laboratories? CAP has an FAQ document out on their website. So if you are a CAP accredited institution, you're able to get into their eLab Solutions suite, and you'll be able to search for this particular FAQ document. My laboratories found it really helpful for them as we were trying to interpret what CAP might be asking for or expecting to see when they come to inspect on this new standard. So I pulled out some of the high points from this FAQ document. So CAP is saying that selecting a general range of just 10 to 80% humidity for the entire lab area is not necessarily adequate. You need to go through your manufacturer defined humidity specifications. And unless otherwise specified, once daily humidity monitoring is adequate for days that the test system is in use. The selected humidity monitoring range needs to be adequate to meet the needs of all test system components used in the monitored area. Things that we've suggested within the labs that we support is that if it's a very large lab space and maybe there are multiple test systems being operated within that area, that they're getting a humidity sensor near to that item so it's as accurate a reading as possible rather than having it on one end of the lab, but testing is also occurring on the other end of the lab and you might not know what the humidity is over there. We're also coaching our labs that they would want to choose the most conservative range for humidity. So if maybe you've got 3 different test systems that are operating within your laboratory, one has a range of 20 to 80% relative humidity, whereas another one has 25% to 79%. We've seen that before. You're choosing what the tightest range is, and then if something were to go into alarm, a humidity sensor goes into alarm, then you can make an assessment of which items within this room are impacted by this low reading. Are some of my items still okay? The next bullet there in that center column, if different test system components have more stringent manufacturer specified humidity ranges, selecting the most conservative range is recommended to ensure notification of the out of spec conditions. And then CAP was clear to say that this only applies to FDA cleared and FDA approved test systems with manufacturer specified humidity ranges. For lab developed tests, the lab is responsible for establishing the environmental conditions needed for testing. And to give you an idea of what this document looks like, I'll just pull this over onto the screen here for a moment. It has a list of 10 questions that CAP thinks would be commonly asked by a lab who's trying to assess whether or not what they're doing in terms of humidity monitoring and out of range humidity responses is going to be viewed as adequate by them when they're coming to inspect. And I'll move this back out of the way. And we'll move to the next slide. All right, so why do environmental conditions matter? Why are we monitoring? Why does CAP care? Why does CLIA care? It's because temperature and humidity levels can impact instrument performance and longevity. High humidity can cause condensation on circuit boards, and that can lead to corrosion and electrical shorts. We care about temperature and humidity levels because it can impact test accuracy and reliability. Low humidity during the winter, oftentimes we're experiencing that here in Minnesota. It gets really cold outside. We have to turn the heat up inside. It causes the labs to dry out. So low humidity can cause static buildup and discharges, which would then impact the volume of specimen or reagent being dispensed in some of our test systems. So it's important to be aware of those risks. And then also specimen or reagent integrity. Improper temperatures can cause degradation or improper reactions. Next, I'd like to share a high humidity vignette to help put this into perspective of what this might look like if you were suddenly faced with a high humidity excursion. How would a lab respond? So in this instance, a laboratory experienced unusually high humidity conditions that in combination with the temperature led to observable condensation on some equipment. This led to unexpected instrument shutdowns and interruptions to testing. The facilities and system engineers that were helping to support the lab through this experience requested the manufacturer's humidity and temperature specifications for all of the impacted instruments. So the lab team had to search for the owner's manuals, the instruction manuals. Some of them had older instruments, and so it was still paper-based. Many of them were newer instruments, so they were able to identify where those PDFs of the owner's manuals were stored. But it was an unexpected use of FTE resources to find that relevant information in the owner's manuals. Some of the manuals that they found provided by those manufacturers didn't feel clear to the folks reading them regarding what the expectation was around humidity or what the risk was to instruments that were being used above their humidity optimal range. So, absent clear guidance, the laboratory had to contact a handful of those manufacturers directly to understand what the implications were to the instrument when they were faced with these high humidity conditions. Then the labs needed to interpret and decide what their next steps were. So some of those owners' manuals just said that it was an optimal temperature range, not a required operating range. And so in those instances, they were saying, what might happen if I continue to operate this piece of equipment, this instrument, in a humidity situation that is above what your optimal humidity range is listed as? And so what they learned is that this might impact the longevity of the instrument. And then the lab and the lab director had to decide what was the path they would take from there. Would they continue to test because the accuracy of the test was not being impacted, but knowing that continuing testing at these high humidity ranges might shorten the life of that instrument? And we know that laboratory instruments are really expensive, and we don't want to have to replace them sooner than we otherwise would have had to. They wanted to be very sure that it wasn't the accuracy of the test being impacted because they felt that if it was the accuracy, they would immediately halt testing because they won't compromise the accuracy of the tests that are being released because we know that patients are being treated based on the answers or the results that are being released to them. Additionally, one of the companies shared with the lab that they recommended to halt testing because there was an increased risk of electrical shock if you were operating at high humidity conditions. And then there's condensation on the electrical circuit boards, and then that could lead to electrical sparks and shocks for the operator. All right, so then we made it through that instance. Obviously, we don't want to have to go through that again. It was a heightened sense of urgency to be able to find the answers and make the right choices. So how do we prevent that feeling of urgency from happening again? We put together a team of lab experts, and they created a tool called the Environmental Conditions Risk Assessment. We lovingly refer to it as the ECRA tool. It's a SharePoint-based database that provides a standardized and streamlined and transparent process for accessing the corrective action plans. So the idea being that the labs are able to enter the manufacturer identified high and low temperature operating ranges, high and low humidity temperature ranges, and then they're able to take from that owner's manual what it is that the risk that has been identified by that manufacturer for continuing to operate outside of those ranges. And then if they do go outside of those thresholds, what is the action plan, the corrective action plan? Are they going to halt testing, move testing to a different lab that does the same testing, that type of a thing? But it's all in one place that all of the labs can access. And this is being rolled out in phases at our institution. So right now we have equipment information and location information that we were able to obtain from our institution's biomedical engineer team. So we really focused in on the testing instruments first, and phase 2 is going to roll in facilities dependent equipment. So thinking refrigerators, freezers, that type of thing. All right, so a little bit more about the ECRA tool. We've got some stats on the side there. Currently, we have 3,177 unique equipment entries, all organized by location. And we have 642 different rooms that have been assessed for the equipment that are within them. We are able to put in the instrument manufacturer model, humidity range, temperature range, the manufacturer's identified risk for environmental condition excursions, and what the lab is going to do about it if we were to go outside one of those thresholds. Some common response plans I have listed here in these bullet points. So some labs will stop testing until the conditions return to an acceptable range. Some labs will send testing to another lab at Mayo that performs that same test. Some labs will continue testing, but increase the amount of QC that they're performing. And some labs are choosing to consult with their lab director for guidance on a case-by-case basis. Some of the benefits that we've realized from the ECRA tool include that this ECRA tool is serving as a reference for looking up temperature and humidity ranges for specific instruments. And it streamlines access to environmental condition information obtained from multiple different equipment or instrument manuals. We don't have to put ourselves in that position again if we were to get into a high temperature, high humidity, low temperature, low humidity situation again, where we have to find all of the manuals, read all of the manuals, and then assess what to do. We've already done it. Our labs are, just slowly rolling through this as they have time so that it's not happening in an emergency. The ECRA tool is centralized, meaning that labs that have similar pieces of equipment or similar instruments are able to see what the other labs chose as their action plan. It doesn't mean that it has to be their action plan, but they can get an idea of what other areas are doing and see if that makes sense for them. We have an automated remote monitoring system, a temperature monitoring and humidity monitoring system that goes into alarm if these thresholds are exceeded. And so this ECRA tool is not replacing that. It's not replacing our lab event management system. So if we have one of these excursions and it causes us to halt testing, we're still going to be recording that in our lab event management system. The ECRA tool is really just meant as a repository for information and initial action plans that have already been pre-decided. And then I already mentioned the last bullet point item here, which is that the ECRA tool is fostering collaboration by providing transparent access to corrective action plans for similar instruments across different labs. And it's allowing for information sharing regarding risks and mitigation strategies. We took some time to brainstorm about how the ECRA tool might support our CAP and CLIA compliance, and we felt that the ECRA tool was demonstrating an awareness of manufacturer requirements, that we've done a proactive risk assessment, and that we've already considered what our action and mitigation plans might be when these out of range conditions are encountered. We also think that it is going to enable us to have faster response times during these temperature or humidity excursions. It's going to lead to consistent practices across shifts because it's the same action plan of halt testing or send to a different lab or continue with additional QC during the excursion period, so that all of our shifts are going to feel empowered to make the same choice, the right choice. And having all of this information in one place, when we've had to ask our facilities team for help with better management of our temperature and humidity conditions within the labs, it's allowing us to have just proactive discussions with facilities HVAC engineers. They can see what it is that we need to meet, and then they're able to get to their response faster about how they might help us meet these environmental condition needs. I have another scenario to share with you. So in this case, we're going to think about the winter when things get dry. So your scenario is that the laboratory experiences humidity levels outside the test system's allowed range each winter when increased heating causes low humidity levels. So the first thing that you would want to do if you were the quality or technical specialist, maybe the supervisor of the lab that is experiencing this, is that you would want to determine if the humidity excursion is occasional, frequent, or consistent. I wanted to bring your attention to the fact that CAP has a CAP Today article in their January 2026 edition. And that article states that if the humidity consistently exceeds the range, then your testing is no longer considered PMI or per manufacturer's instructions. And so for those of you who get the slides after this, that's a hyperlink out to that article, and that is a public article. You'll be able to read it. We found it to be really interesting to get an insight into the way that CAP was thinking about this new humidity standard that they have. CAP also in that article has some suggestions for what you might do as you're trying to figure out what to do about this humidity level. Is it something that's happening all the time, or was just a quick blip and then it got better? So you could contact the manufacturer to determine if the humidity range can be expanded. We did do that. There were a few instruments from one specific company where they said, yes, we can actually expand that range. And then they gave us a technical bulletin that indicated that, because obviously they're not going to print a new or create a new owner's manual for us. But they did give us a technical bulletin that gave us that expanded range. And so some important questions that you could ask them would be, did they set that range for upper and lower humidity for that instrument? Because that was just the limits of that manufacturer's setup. They just couldn't get it to be more humid as they were testing. They couldn't get it to be any drier in that area. Was the range set up because they started to notice that testing performed differently, not as accurately outside of the ranges that they set? In that case, if it's impacting accuracy, reliability, or precision, those ranges are pretty hard set. Once you have a better understanding of why the humidity ranges were set where they were, and if they are not able to be expanded, then you're encouraged to seek help from your facilities team. So you could ask them to help you better understand what is your lab's humidity setting? Can the building automation system be updated to increase humidity if your lab is too dry? Could you have a dehumidifier added if your lab is too humid? And is the humidity sensor in an appropriate location? And your facilities team can help you with that too. So things that you would want to consider is the humidity sensor installed near a path of travel, a walkway within your lab? Are you getting a lot of movement back and forth that's creating like wind or breeze, right? So things like a doorway, you'd want to move your humidity sensor away from that so that airflow pattern isn't impacting your humidity readings. Is your humidity sensor put near an exterior wall? Is your temperature sensor in direct sunlight if you happen to have windows within your lab? So those types of considerations can be made. And if you don't have your sensor in an optimal location, you can ask the facilities team to help you get that moved to a place that is more appropriate, less impacted by the movement, the sun around it. All right, so I'd like to take this opportunity to share with you what the ECRA tool looks like for us. I will say that this took us about a year to come to fruition when our team came together to try to figure out how we want it. So we thought about this before CAP updated their checklist item. It was just something that we wanted to have just get our arms around a little bit better, feel a little more comfortable with knowing what our upper and lower ranges were. We had considered just doing a simple like Excel spreadsheet type document that each lab would just maintain on their own, and everything would kind of be separate and not viewable to each other's labs. As we dove into it a little bit more, we found that there would be a benefit from having it on this SharePoint so that more people have more eyes on what's going on within each lab. So that is how we ended up with this document. Oh, let's see if I can get it to expand here. All right, so this is our landing page for the environmental conditions risk assessment. We have a general purpose statement and then some quick links out to various support requests or training. But really how the labs are interacting with this are on these three views or tabs: the environmental conditions risk assessment tab, the equipment models tab, or the work areas tab. So looking at the environmental conditions risk assessment tab, we have all of our work areas available as they log in. They're certainly able to, this is taking a while to load. There we go. So we, hold one moment while it loads. Perfect. All right, so the users are able to sort by their physical location or by their work area name. And so one of the examples I'll show you here is for our bacteriology area. I don't have this set to have a certain view, but one of the benefits of using SharePoint is that you as a quality, a technical, a supervisor likely are only responsible for one lab or maybe one lab, but that has 2 or 3 physical locations. So you could set a custom view so you're really only seeing your items when you come in here. In my use case for this, I am interested in seeing everyone. So for today, we'll show you what bacteriology looks like. And we have all of their instruments loaded in that we were able to receive just as a data share from our biomedical engineers. We know what building they're in and what physical room within that building they're in. And so many of our labs span several different rooms. Each of these rooms has temperature and humidity monitors. And then we identify high and low humidity, high and low temperature, and what the risks are if, per the manufacturer's manual, what it is that might happen to that instrument or to the accuracy of the test if it were to go outside of those defined conditions. So looking at, let's look at this one specifically, the back tech. As we click on this, we'll get a new window that opens. We can see where it is, who owns it, the lab's humidity risk assessment, what their plan is if it were to go out of range, and the same thing with the temperature risk assessment, what they would do if it were to go out of range. This is not the place to record information about an actual excursion. That's in a different system, the monitoring system, the event system. But this is your repository for what do we know about this piece of equipment. So one scenario I'd like you to consider is you're a technical specialist and you work in the bacteriology lab, and your temperature and humidity monitoring system, that system is giving you an alert. So you go and you figure out which room it's giving you an alert for. So in this case, let's say it is room 462A, and it's telling you, your monitoring system is telling you that you have a low humidity alarm, and it is 22% humidity in that room right now. So by using this tool, you'd be able to figure out which items within that room are going to be impacted by a humidity of 22%. So I'm looking at my low range, and if my humidity goes to 22% and these two items tell me that they have a low range of 25%, I can see that these two are being impacted by this humidity reading that our monitoring system is giving us. I want to know what it is that I should do as a person within this lab. Can I continue testing? Is it safe? And I can see that the assessment for this piece of equipment is that they're going to continue testing and monitor closely. Something within their test SOP might allude to the fact that they'll run additional QC, or what would happen if after continuing to monitor closely, they do begin to see errors within the test or the run errors out, then what the next step is. Okay, move that one away and we'll come back over here to the presentation. I wanted to leave you with some of the lessons that we learned as we were rolling out this ECRA tool. So we started with a slow rollout. We did like a test or a pilot group so that we could get some feedback from some of the users about the functionality, the ease of operations, the accuracy of the information that we were getting in from our biomedical engineer team. And then once we felt like we had a solid SharePoint, then we were able to open it up to the rest of the labs and have a relatively smooth rollout with it. So as lab instruments, equipment, and test systems were evaluated by the labs to fill in their ECRA workbook, it became apparent that our humidity fluctuated over the course of the year. And so we've got this automated remote monitoring system, right? So we're watching temperature, we're watching humidity. We have the ability to see the trends over time. And we saw that in the winter, with increased heating needs inside due to cold temperatures outside, the labs tended to be drier, so having lower humidity than they did during the summer months. And having that continuous monitoring of these environmental conditions within the labs really helped us understand where we might be at risk for going outside of the thresholds that were set by the manufacturers. Some of the impactful variables that we found were contributing to the humidity trends that we were observing was the building that the lab was in. So was it a newer building or an older building? Meaning, does it have a newer building automation system or an older HVAC system? What is the external cladding of the building? Is it granite? Is it stucco? Is it brick? Are there a lot of windows? All of that was impacting the ability of the lab to hold appropriate temperature and humidity over the course of the year and the fluctuating temperatures outside. We also found that the location of the lab within the building itself might play a role in their ability to meet temperature or humidity needs, meaning that we found differences between interior rooms, exterior to the, like along the outside walls, or in the basement, like lower levels, that type of a thing. We also were interested to find that some of our labs that have a higher number of people and instruments working within that lab space also struggled a bit more with temperature or humidity, staying within the thresholds that were identified by the manufacturer. And then trend graphs from our automated remote monitoring system were helpful when presenting these challenges that the labs faced to the facilities team when asking for assistance with improving control over the environment where testing was being performed. Some takeaways for you as we're winding down on our presentation here today. Just highlighting again what changed, that CAP retired their Lab General 61300 climate control checklist item, and they have a new CAP All Common humidity specific checklist item. The importance of this is that the content of the standards were similar, but when it was moved from the Lab General to the All Common is now making it visible to more inspectors. And so we are anticipating an increased focus on test systems that have specified humidity ranges. And we're hoping that this ECRA tool that we've put into place is going to help us be proactive and more in control of knowing what our humidity and temperature needs are, and then also identifying what our action plans, corrective actions will be if we experience these excursions. I'd like to suggest a practical approach by testing labs. So I think it's really important to get familiar with the instrument humidity and temperature requirements for the items that are within your lab. You should assess your current ability to consistently meet those requirements. So CAP used that word, consistently exceeds. So kind of, pulling from that is how often, how frequently are you being able to keep your temperature and your humidity within the defined ranges for the test systems that are operating within your laboratory. I would suggest performing a risk assessment to determine risks of not meeting environmental condition requirements. So consider accuracy, safety, and instrument longevity. I can't encourage you enough to partner with your facilities group to find a solution. When we first started in on this journey, we didn't have the right contacts within our facilities group. But each time we sent an email, made an inquiry, we eventually found the right people to partner with, the people that were able to take our needs into consideration and affect change within that building automation system to help us make the easy changes, right? We can't change the external cladding on some of our buildings, and that's going to be a longer term fix. But for some of those quick wins, just finding the right partner, that was the key. And then be mindful of the possibility of having to revalidate your test as an FDA modified test if the manufacturing operating conditions cannot be met. And obviously that's not ideal. That's not something any lab wants to have to do, but that is something that CAP highlights within that FAQ document for the humidity checklist item. That is something that might have to be considered if you just can't get your humidity within range. And that brings me to the end of the formal presentation. So I'd like to open it up for questions, comments, and discussions. And I think I have the ability to open up the Q&A. And it looks like we have a few. All right, our first question is, previously when humidity was too low, we would document that QC was acceptable and therefore patient testing was not affected. This was clearly documented and in our policy. However, this was deemed insufficient by CAP and we were cited. How would additional QC during times of low humidity be considered acceptable? So if we're in the middle of, we haven't been cited by CAP yet. Let me start with that. We don't know how this answer will hold up. Our approach to this is we only have a limited amount of sample. If we were to stop testing in the middle of the run, there's a chance that we won't have enough sample to continue that testing. The importance for us is the needs of the patient come first. We want to make sure that we are able to deliver accurate and timely test results. And so if QC is showing that that test worked as expected, we plan to report the test. Also, CAP was clear on saying that if it consistently exceeds its ranges, and maybe this was a 20-minute blip where we were a little bit too dry, we think that we could reason that it was acceptable to continue testing versus if it's the winter months and we know for the entire month of February, every day that we do testing, it's going to be 15% humidity in the lab, but we need to have a minimum of 20%. We would not be able to justify that with increased QC. That would have to have a different answer. I hope that maybe lends some approach to the way that explanation to the approach that we're going to take should we be challenged with it. The next question on the list is, how do you handle high humidity conditions for clinical samples stored on the lab bench? So being that we're in Minnesota, we don't generally experience high humidity. Occasionally in the summer, there may be an instance where things get a little too warm within the lab. But again, it's never for it. It would have to go above 80%, and that's just not likely to happen in Minnesota. I can see that that might be something that would be something harder to control if you were in a warmer, more humid climate. So I don't have a good answer to that one. Perhaps the answer is to not store those samples at the lab bench, so keeping them refrigerated until it's time to use them, only bringing them out when it's time to bring them to the right temperature. The next question is, does the humidity requirement apply to glucose strips located in a central supply area outside of the lab? Yes, and also, are those glucose strips contained within some sort of a package that the strip itself is not impacted by humidity or temperature until the package is opened? That could help you. If they are open and exposed to whatever the environment is, then I would say maybe you don't want those strips outside of a lab or outside of an area that's not being monitored for temperature and humidity. Let's see, our next question. For microbiology incubators, how do we find out what the range of humidity is required? We used to have 80% as the upper range, but we have a new incubator and have no way to lower it. We place water in the lower tray of the incubator and it goes to 90%. Cannot say for sure besides checking with the manufacturer. If the manufacturer does not have an upper limit, then you don't have to manage an upper limit. If the manufacturer has an upper limit, then there's got to be a way to work with the manufacturer to have them hopefully give you hints, suggestions, tips for how to use the product that they sold to you. The next question is this tool available for use at our facility? So I'll say no right now. It's not something that we intended to share externally or put for sale on the marketplace. We shared it as an idea for what you might be able to do, maybe if you've got someone that's handy with SharePoint or if you've got IT resources at your facility, to help you come up with something that was more of a living document than just an Excel spreadsheet. However, that is where we first thought we would start, is just with a spreadsheet specific for that lab and let each person or each lab manage it on their own. So I think with the right resources, you might be able to develop it. Laboratories outside of DLMP, can they access the SharePoint? So we are making this available to share across the various Mayo sites. Right now it's specific to Mayo, Rochester, and the DLMP labs. We're slowly starting to bring in the health systems. However, if you're at Mayo and you're in a non-DLMP lab, you can reach out to me and we can work with Amy Robinson and we can find a way to get your lab's inventory in there as well. And we'd be happy to roll you in. All right, let's see our next one here. At what point would we need to consider moving room temperature products if the temperature falls out of range? Great question. That is up to your lab director ultimately, but you would need to determine, is it a half an hour? Is it 20 minutes? Is it immediately that you move things? There's no lab general or all common or CLIA lab general requirement for how long something can be at the wrong temperature or wrong humidity before it has to be moved. If you are in a specialty area like transfusion medicine, immunohematology, I think histology might, there is a bit more guidance about how quickly things need to move. But for general labs, it's up to the discretion of the lab director ultimately. All right, our next one for micro, too low would cause media plates to dry out. Yes, definitely. Too high might cause condensation on the plate. Yes. How do we define the upper range? So if you're purchasing those media plates, I am hopeful that it comes with some sort of a package insert, for use instructions, something like that. If you're creating your own media plates and it's being used within a test system in your area, ideally this would be defined during your validation phase, that your test developers or your technical specialists have identified what is your low end of humidity and what is your high end of humidity, so that you know that the resources that you're using to produce the results have a range at which they can be used at. So the answer would be, how do you define the range? Either through the manufacturer's information or through data that you've collected from the validation test runs that you've done before putting a test into use in your laboratory. We have another item here. I can't find anything in micro cap checklist which defines humidity. So it's only by manufacturer of the incubator. Correct. Yeah, if you don't have anything in your specialty checklist, then manufacturer's owner's manual, service manual, whatever that might be, that's what you would go by. If the owner's manual doesn't say an upper or a lower range of humidity, then there isn't one from the manufacturer, but we really encourage labs to include that within their validation plan. You'll know what gives you accurate, safe, reliable results within your own test system, where it's going to be used within the room that you're going to be in, the building you're in. So defining it yourself is the way to go if it's not defined for you. That's what I would encourage. Have you implemented humidity monitoring in storerooms that supplies and reagents are kept in, but no testing is done on instruments? I'll say yes and no. Some of our areas have implemented those humidity monitors there. That is not ubiquitous across all labs yet. That is something that we're working for. We obviously identified that that would be a best practice suggestion. There are some labs that have had that in place for years. They are monitoring temperature and humidity of an enclosed storeroom, so they know exactly the small micro environment of that room, so that they know that when they go in there to get that supply, that it's been stored appropriately before they put it into use. So again, best practice, not all labs are doing it, but encourage it as a direction to move in.
Awesome. Thank you, Lisa. I'll give a final call for questions. So if you have one on your mind, please put it in the Q&A box here. And we want to thank everyone for participating in today's webinar. Again, I'm not seeing any further questions. If you have any questions that you think of after we end up here today, you can go ahead and include them in your evaluation, and you can access the link to that evaluation in the chat now. Laboratory credit is extended for this program, and upon finalizing your evaluation, you will have the option to claim credit and print your certificate. Also, as a reminder, this program was recorded and will be posted on our website in a few weeks. Thank you everyone for joining us, and have a great day.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.